Hi everyone, hope you all are good and fine. So as I was previously mentioning in my previous class that about the glycolysis, and after the glycolysis the process called the Krebs cycle. Okay, so you will think that where this cycle happens. This cycle happens into the mitochondria of eukaryotic animals. Eukaryotic means the advanced cells, and the cytoplasm of the prokaryotic cells. I will not take so much time to make you understand about the Krebs cycle. You will automatically understood. Look, this is the pyruvate. Pyruvate is the end product of the glycolysis, and it is a 3-carbon compound. Yes or no? So this pyruvate is converted into acetyl CoA, and this acetyl CoA is the intermediate product between the glycolysis and Krebs cycle. So acetyl CoA is a 2-carbon compound. That means here also 1 carbon dioxide is released. Yes or no? So we can say Krebs cycles and the glycolysis pathway are interlinked via and molecule. That intermediating molecule is the acetyl CoA, and it is a 2-carbon compound. Now after the oxidation, this acetyl CoA mixed with the oxaloacetate by the help of citrate synthase enzyme to make the citrate. Okay, citrate, it is a 6-carbon compound. This citrate is now converted into cis-aconitate, then isocitrate via the activity of the aconitase enzyme. Now you will think, sir, why we should remember these too much names? This is a cycle, boys and girls. So you have to remember this name in order to memorize the things. First it was the acetyl CoA. Acetyl CoA, it's mixed with the oxaloacetate, and then it forms the citrate via the enzymatic action of citrate synthase. Then citrate is converted into cis-aconitate. Cis-aconitate is converted into isocitrate. Okay, it was okay, and all the things were 6-carbon compounds. Isocitrate, it was also 6-carbon compound. Now look, the name isocitrate dehydrogenase. Dehydrogenase means the activity of NADH. Okay, just think about the glycolysis pathway. There was glyceraldehyde 3-phosphate. The enzyme was glyceraldehyde 3-phosphate dehydrogenase. This was NAD plus to NADH, the reduction of NAD. NAD means that molecule, nicotinamide adenosine dinucleotide phosphate, like that, NADH. So now NAD plus is converted into NADH. Okay, via isocitrate dehydrogenase, and also 1 molecule of carbon dioxide is released. Very important process, very important step, that via isocitrate to alpha ketoglutarate 1 CO2 is released. Now if you think that if 1 CO2 is released, then how many carbon will remaining there? There was 6 carbon, 1 carbon gone outside. Then alpha ketoglutarate is a compound made up of 5 carbon. Okay, again you can see the name of the enzyme, alpha ketoglutarate dehydrogenase. Dehydrogenase means again the NAD plus will convert it into NADH. Okay, NAD plus is converted into NADH, and also 1 CO2 is outside. So we can say that succinyl CoA, it is a 4-carbon compound. Yes or no? It is a 4-carbon compound. Via succinyl CoA, it goes to the succinate. The succinate, that enzyme called succinyl CoA synthase. Synthase, remember synthase is what I said in the previous class. ACE means ATP or energy formation. So here one thing will release in the form of GTP. GTP will produce. Okay, not ATP will produce, here GTP. Okay, so succinate to fumarate via succinate dehydrogenase, again dehydrogenase. So genase means, now here there is another currency. Look, ATP was a energy currency, NADH is an energy currency, there is another energy currency. The name of the energy currency is FADH2. Okay, the full form of FAD is the flavin adenosine dinucleotide phosphate, like that, FAD. Okay, that is also an energy currency. So here also that FAD plus will reduce into FADH plus H plus. That means here is no NAD, here is FAD. Okay, then fumarate is converted into malate via the enzyme action fumarase. Okay, and malate is converted into oxaloacetate, and entire these things are 4 carbon. Obviously I have said 4 carbon because it has to be 4 carbon so that it can add with the acetyl CoA to make the 6 carbon compound, that is citrate. Yes or no? Yes or no? That is why that malate is now converted into oxaloacetate. Don't think about that, sir, I cannot remember these names. You can remember. Yes, citrate, cis aconitate, isocitrate, alpha ketoglutarate, succinyl CoA, succinate, fumarate, malate, and after that it was oxaloacetate. I can remember, you can also remember. Easy. If you go through 1 to 2 times, you can remember. Okay. So this oxaloacetate, it is a 4 carbon compound, but by which enzyme it is converted into oxaloacetate? Malate dehydrogenase. Again dehydrogenase. Dehydrogenase means again NAD will be converted into NAD plus, will convert it into NADH. Okay. So as I earlier mentioned, via glycolysis what we get? 2 ATP. Yes or no? It was about the glycolysis. It was the glycolysis. We get 2 molecules of ATP, or if we consider that NADH, then it becomes 8 molecules of ATP. In the previous video I have already mentioned. But in Krebs cycle, how much amount of energy, what are the things we are getting from Krebs cycles that is happening within our mitochondria of eukaryotic cells? Look, let us count together. So try to observe this box very carefully. How many NADH molecules are there? 1, 2 and 3. Yes or no? So I got 3 NADH. Yes or no? Yes sir. Okay. How many molecules of carbon dioxide I get? In this step, 1 CO2 gone, 6 CO2 becomes 5 carbon, and 5 carbon becomes 4 carbon here. So 2 CO2 is produced. How many molecules of GTP? 1 molecule of GTP. How many molecules of FAD? 1 molecule of FADH. So this is the product I am getting via a single cycle of Krebs cycle. Here is also a question, because this is not for the junior class, I will not say. This is for the 11, 12. Na? So you have to be very careful, because after that you are going to appear for the NEET examinations. So here these kind of basic things are very much important, and you have to memorize all the small things, because questions come from very small minute area. So this is the entire thing that is gone for a single pyruvate molecule. But think about what I mentioned in the previous class, via glycolysis you get 2 molecules of pyruvate. Yes or no? 2 molecules of pyruvate. That glucose is 6 carbon, pyruvate is 3 carbon, we get 2 molecules of pyruvate. That means this entire thing will be multiplied with 2. So via Krebs cycle, what is the final product? We are getting 6 NADH, 4 CO2, 2 GTP and 2 FADH. Is it clear? This is the net gain of the Krebs cycle. Okay, hope I have said not little fast, I won't say, but I think this is the Krebs cycle, boys and girls. You go through it, it is very important. Okay, next, in the next class I will talk about electron transport chain. Okay, and ultimately we will derive that equation how via 1 molecule of glucose we can get 38 molecules of ATP. Hope it is understood to you, and thanks for watching, and stay tuned for the updates. Thank you.